see is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
So there's a nice 3D process happening for transcription. So there are some questions about the regulatory sequences when we were talking about getting the RNA, the DNA transcribed into RNA. So you saw when we were making the house that there were some elements that were common throughout all the rooms and some elements that were only common to a few rooms or just to specific to a single room. So same thing within your cells. There are some genes that need to be expressed in every cell of your body. There are some genes that need to be expressed only in specific types or only in a single type of cell. And so the information contained here, what I'm calling the regulatory sequences or the promoter. That's DNA sequence that the cell can understand to know when a gene should be expressed or when it should not be expressed. So that's what I was referring to when I said the promoter or the regulatory sequences here. It's just the information that the cell needs to know whether or not a gene should be expressed in that particular cell type at that time. So I know a bunch of you had questions. Are there more questions? Yeah.
So maybe I'm dumb.
Probably not.
No, I am dumb. But so the promoter is a type of regulatory sequence.
The promoter refers to the stretch of DNA that is ahead of the first exon.
Okay.
Which is the right person. And those are different things.
I'm going to use those words interchangeably.
Okay.
But it's
So are like all promoters regulatory sequences and all regulatory sequences promoters?
There are sequences of DNA that the cell recognizes, and they are found in this region compared to where the actual protein coding sequence is.
Okay. So what's so regulatory about that sequence?
The cell recognizes specific sequences to know whether a gene, should this gene be expressed in skin cells or should it be expressed in heart cells? So the sequences here the cell recognizes.
And then depending on the depending on the protein, it will lock on, certain ones will lock on to certain regulatory sequences, and then they'll copy those because they're going to be bringing that to the right
Oh, wait,
place.
wait,
So the transcription factors recognize these sequences, and transcription factors call RNA polymerase over.
Oh.
Sorry. So there are proteins in the cell that recognize those sequences, and then those proteins call RNA polymerase to have the gene transcribed.
Makes sense.
And that's the relationship between the two. Okay, that makes so much
But I'm gonna, so I'm sort of using this interchangeably, because this is where regulatory sequence occurs, is in this region just upstream of the actual protein coding gene.
Okay.
Are there other questions about promoters, transcription factors, RNA polymerase? Okay. So where we left off, this gene was transcribed into RNA. So now let's take a look at getting our RNA sequence translated into protein. So here's our mRNA. So the first thing we need to think about, actually, so we know that DNA and RNA were both 4-letter languages, but the amino acids that are part of the protein, there's 20 of them. So how do you go from 4 letters, A, U, C, and G, to a 20 amino acid language that makes up proteins? So we can think about whether it takes, right, if it was just a 1 letter, 1 amino acid, how many combinations, how many words could you make? How many protein, or how many amino acids could you code for if it was that same 1-to-1 that we had with DNA and RNA?
a lot.
4?
One amino acid, one.
So RNA has 4 letters, A, U, C, and G.
Yeah.
But there are 20 amino acids that are encoded for by the genome. So if you had that same, right, we're going from DNA to RNA, if there was an A in the DNA, you knew that it was a U in the RNA. If it was a C in DNA, you knew it was a G in RNA. But now we're going from 4 to 20. So if it was just based on 1 letter at a time, you'd only be able to have 4. What if you had 2-letter words? 16 combinations. What if you had 3-letter words? 64, and then 4-letter words?
218. 36.
4 times 4 times 4 times 4. But anyway, so what if those makes the most sense? 1 and 2 certainly can't possibly make sense because you're not coding for enough amino acids. 4 ends up being way, way, way too many. So obviously 3, right? So there are 64 different combinations to code for those 20 amino acids.